Hello students, welcome to the new lecture, and in this lecture we are going to start with the new chapter, okay, ninth class, okay, biology, okay. So this is a new chapter, that is chapter 4, transportation through plasma membrane. So this is a topic that we are going to discuss in this lecture, okay. So first we are going to discuss about what is plasma membrane and how it is important and all those things that we are going to discuss now, okay. So here the cell is a structural and functional basic unit of life. What is meant by structural? It means a cell has a particular structure. Each and every cell has a particular structure. Okay. And by the way, good afternoon to everyone. And if you have any doubts or questions, post it in the comment section or live chat section so that I can see and also I can answer to your question or doubt. Okay. So what is structural? Structural means it is going to have some structural. Each and every cell has some kind of structure. In the last class, what we have discussed? We have discussed about striated, okay, non-striated, like cardiac muscles. Okay, they have the branches. Okay, they have the branches and so on. So these are the cardiac and so on. So these are the different types of structures. It means that each and every cell is going to have some structure. So they have some kind of function. What is function? Example, if you take heart cells, the heart cells is going to have some kind of a function. Okay. So next is a basic unit of life. It means each and every cell is a basic unit of life. It is mainly responsible for the survival of an organism. And again, we have discussed that the cells. Okay, the cells are of two types. What are the two types of cells? Okay, they are unicellular and also multicellular. What is unicellular? Uni means only one cell. Means if organism is made up of one cell, it is known as unicellular. What is multicellular? Okay, already we have discussed all these things. What is multicellular? The cell containing many cells. If organism is made up of many cells, it is known as multicellular. Okay, and here all organisms is made up of cells. Okay, whether it may be any organism, whether it may be human being or animal or bacteria, they are made up of cell or cells. Okay, so the cells are going to allow only certain substances to pass through them. It is known as selectively permeable. So what is selectively permeable? It means the cell is going to have cell membrane. So this is a cell and also cell membrane. What is cell membrane? It is outer covering of the cell. Okay, cell membrane is nothing but outer covering of a cell is known as cell membrane. So inside there is a new cell, nucleus. Okay, so Golgi apparatus, endoplasmic reticulum, Golgi apparatus, and so on. It means here the outer covering is a cell membrane, and this cell membrane is going to allow only certain substance and will not allow other substances. Means it is selectively permeable. How it is selectively permeable? Selectively means the cell membrane allows only selected components or selected substances to pass through it. So this is the reason we are calling the cell membrane as selectively permeable. Okay. So here we have some activity. We have one activity. This activity is get in out of the cells. Okay. In this activity, we have to find out what are the substances that enter into the cell and what are the substances that go out of the cell, that we have to discuss over here. Okay. Here the substances here. The substances are many types. So the few substances are given. The oxygen is given, glucose is given, proteins, everything is given. Okay. So which can go into the cell. For example, if you take oxygen will enter into the cell. Okay. Yes, it is going to enter into the cell. So can go out of the cell. So oxygen will not go out of the cells. Okay. Oxygen enters into the cell. So glucose, yes, the glucose enters into the cell and can go out of the cells. So no, glucose cannot go out of the cells. So next is protein. So can protein go into the cells? So all proteins will not go. Okay. So proteins cannot go into the cells and also proteins cannot go out of the cells. But some proteins like antibodies, they will move out of the cells. Okay. So yes, the proteins also move out of the cells, and in some cells the proteins will enter into the form of amino acids. Okay. Next, fat. So fat will not enter into the cells. Okay, generally will not enter into the cells, but in the form of a fatty acid, they will enter into the cells. Okay, so can go out of the cells. So generally the fat sometimes it can go out of the cells. Vitamins again same. So it means these fat proteins, fats, vitamins, they are in complex in nature. Okay, they are complex in nature. So when these nutrients digest, they will turn into amino acids. Okay, next they will turn into fatty acids. They will turn into uh, sugars and so on. So then they can enter. Okay, then they can enter. So when these nutrients are in complex shape, they cannot enter. When they get digested in the form of simple substances, then they can enter. Okay, so next, minerals. As minerals enters into the cells, okay, they cannot, sometimes they will move out of the cells also. Carbon dioxide cannot go into the cells, carbon dioxide exit out of the cell. Okay, waste materials cannot go into the cells and they exit out of the cells. Okay, so this is about the activity that we have discussed. We have to discuss. Okay. So the cells performs different functions and substances are required for the cells. Some substances are required. So what are the substances required for the cell? There are different types of substances required for the cell. Example, solids. Okay. So solids are required for the cell. Example, glucose. Glucose is essential for the cell. Next, liquids also are important. Liquids. So liquids like water. Water is essential and this water is important for the cell. Okay. Gases like the gases. What are the gases like carbon dioxide? Not carbon dioxide, oxygen. Oxygen is important. So it means these glucose or nutrients, water and also gases are very important for the cell for the survival process. Okay. So next is preparing solutions. How do you prepare solution? First of all, what is meant by solution? Solution is nothing but it is made up of solute. Okay, solute plus solvent. Okay, solute plus solvent is equal to solution. So what is meant by solute? Solute example, salt. Okay, salt is example for the solute. Solvent example, water. Water is example for the solvent. If you mix salt and water, what will become? Salt water. Okay, salt water. So the salt water is the solution. Okay, salt water mixed together to form salt water. So the salt water is a solution. Okay, so remember, solute is a solid substances. So they are solutes and they will mix with the solvent, example water, to form a solution. So here the stir well to dissolve the sugar in the water to prepare the sugar solution. So if you want to prepare sugar solution, prepare in this way. Okay. So next is the preparation of a saturated solution. What is saturated solution? Saturated solution is nothing but the solution which is more concentrated and no other substance cannot dissolve in it. Okay. And by the way, if you are able to listen my voice, just type yes. Okay, type yes in the comment section. So if you are able to listen my voice, type yes in the comment section so that I can confirm that you are listening and so on. Okay. And if you have any doubts or questions, just type in the live chat section so that I can read and I can answer to your questions. Okay, just type yes in the comment section so that if you are able to listen, if you are listening my voice. Okay. So we will discuss the next topic, that is the preparing solution. So how do you prepare the saturated solution? So you can prepare saturated solution, okay, so with the help of any solute, okay, excessive mixing of solute, you can prepare saturated solution. So here you can see, so take 100 ml of cold water in a beaker, okay, 100 ml of the cold water. So beaker containing cold water is about 100 ml, okay. So to the 100 ml add some amount of salt, okay. So some amount of salt or sugar. So and stir it, after some time again some more and some more. Add the sugar or salt in that way, and also repeat addition of the sugar and salt and mix it. So after some time, what you will see, after some time you will see that will not dissolve, that is undissolved condition will form. So this undissolved condition will be formed. This is known as saturated solution. What is saturated solution? If the sugars or salts, addition of excess sugar or salt in the water will not be dissolved, and the solution will become more and more concentrated. So that is known as saturated solution. How do you prepare saturated solution? Add excess amount of sugar or salt and mix it, and some amount of sugar will be left over at the bottom. So this is an indication. That saturated solution is prepared. Okay. So here is small activity. It is about which one is more concentrated. So here we have to take the 3 beakers. We have to take 3 beakers, and also in the beakers, in each beaker. Okay, this is one beaker, second beaker, and also third beaker. In each beaker, 100 ml is there. 100 ml of the water will be taken. So in the first beaker, add half teaspoon of the sugar. In the second beaker, take 1 teaspoon of the sugar, and the third beaker, take half teaspoon of the sugar. Okay. So then dissolve it, and also after dissolving, check the concentration, which beaker is more concentrated. So obviously the second beaker containing 1 teaspoon of the sugar is more concentrated than the remaining 2. Okay, it means the concentration is depending on the amount of the solute. Okay, so what is solute? Sugar. Here the sugar. So second beaker is more sweeter in taste. Why? Because it is more concentrated. Why it is more concentrated? It is more concentrated. Why? Because more sugar. Okay, more sugar is added. It means more solute is added to the second beaker. It means we can confirm, okay, the sweetness or concentration of any beaker depends upon the solute added. Okay, solute added to the solvent. Okay, this is about it. And in the next lab activity, we are going to discuss about the different materials in the solutions. Okay, so for this we need 2 beakers. So beaker 1 and also beaker 2. So in the beaker, what we have to do? We have to prepare the solution, normal tap water. In the beaker 2, we have to make saturated solution. What is saturated? If the solution having more amount of solute, okay, it is known as saturated solution. So here, in the procedure, take 100 ml. So take 100 ml of the water in the beaker, and also keep 1 dry raisin. Okay, this is nothing but dry grape. Okay, so keep 1 dry grape. So here, what is happening here? Okay, So here, the dry grape is previously this much, and after 1 hour, it is going to turn this much height. Why? Because it is going to increase its size. How the dry grape is increasing its size? Because it is going to absorb water which is present in the beaker, and it is going to increase its size. The size is increased because of absorbing water. This process is known as swollen. And why it is having like that? It is due to the osmosis. It means, so raisin is in this way, and it is going to absorb water, okay, and turns into this much height. So this process is known as osmosis. What is osmosis? The beaker water is less concentrated, okay, and raisin is, okay, more concentrated. As a result, water moves into the raisin. Water will move into the raisin due to the osmosis. Osmosis is nothing but the movement of water molecules from the lower concentration to the higher concentration. It is known as osmosis. Okay, so by this way you can see the swelling of the raisin. Okay, and leave it for 1 hour. After 1 hour, if you see, the size is increased. So in the second beaker also you can see, what you can see? So saturated solution. So keep the swollen raisin in the beaker. Okay, so here, and wait for 1 night. After 1 night, the size of the raisin is going to decrease. Let us consider this is the size of the raisin. So after one night, the resin will shrink in this way and size is going to decrease. Why it is happening? Why? Because here the solution is more concentrated. Solution is more concentrated. As a result, water from the resin is going to move into the saturated solution. As well, the size is going to decrease. Okay. So this is about exosmosis. What is endosmosis? Okay. What is endosmosis? It means the movement of water in. Okay. So water will move inside the resin, that is known as endosmosis. What is exosmosis? It means the water will move out of the resin. Okay. Water out. Okay. So why the water movement and why the water enters in and exit out? Why? Because it is due to the osmosis. Okay. So here you can compare the resin size over here and the resin size over here. Okay. So this is the process how it is going to occur. And next one is lab activity. In this lab activity, we have to see the osmosis. We have to observe osmosis. So here is we have to take one raw potato, petri dish, 2 pins, water, sharp knife, okay, and so on. So we have first the procedure is take potato and peel off the skin, remove the peel and cut into cube. So we have to make the potato into cube that you can see here. After converting into the cube, make a well, okay, make a small well inside it and also pour some amount of saturated sugar solution inside the cube. Okay, so we have made a well now. This is the potato. We have made a well and inside the well pour some amount of sugar solution and put a pin indicating the level of the sugar solution. So and wait for some time and put the whole setup in the petri dish. Okay, so this is the petri dish. So put in the petri dish containing water that you can see in this diagram. In this diagram, you can see the petri dish. So what will happen after some time? After some time, the level of the water is going to increase. The level of the water which is poured in the well is going to increase. Why? The reason is osmosis. Due to the osmotic pressure, so due to the osmosis, the level of the water in the well of a potato is going to increase due to the osmosis. Okay, so due to this reason we can say that osmosis is nothing but movement of water from the lower concentration to the higher concentration. Okay, and I hope this lecture is helpful to you. If you have any doubts or questions, feel free to